Okay, so this lecture is on the reactions of photosynthesis. Here are the objectives. Make sure that you read over them and familiarize yourselves with them. When you finish the online video, come back, see if you can answer them. If not, you may want to review your notes or watch the online video again. As always, if you have any questions, make sure that you ask your teacher either through email or in class. So let's focus on the big picture here with photosynthesis. We see the equation down at the bottom of this slide. Plants will take in carbon dioxide, water, and light, and from that they will produce sugars and oxygen, or more specifically, glucose and oxygen. So the oxygen is what we use in order to breathe, and we're going to need that for cell respiration. And the glucose is going to be used and broken down to extract energy from in order to make ATP, so our cells have energy that they can function. So we need plants. We would not be able to survive without the products that they make from photosynthesis. So let's start breaking down this equation and start looking at each individual reactant or product and figure out where it is entering the leaf or the plant or whatever it is. And then what we're going to do is we're going to go even deeper and get into the specific steps of photosynthesis to see where each piece is used. So first thing, let's take a look at the sunlight energy. Sunlight is going to be taken in by certain cells of a plant. Specifically, we're going to be looking at ones that are green because they contain the pigment molecules chlorophyll. Let me just write that over here for you. Okay, remember chlorophyll is what causes most plants to look green, and so it is this pigment that is going to absorb the sunlight's energy that we need for photosynthesis. Therefore, chlorophyll is really only going to be found in the green parts of the plant. So, if you have like the stem or like the trunk of a tree, if it's not green, it does not contain chlorophyll, and therefore the cells in that part of the plant are not going to be taking in sunlight energy. So that is why your leaves are very broad and flat, so that they can sort of cup themselves and angle themselves towards the sun to take in as much energy as possible. Then we also have a need for water. Water is going to come in through the roots, so it's going to come in through the bottom of the plant. That's why whenever you are watering, flowers in your yard or something like that, it's much more important to get the water down on the soil where the roots are so it can be absorbed. Plants will not take water in through their leaves. It's not possible. So water is going to come in through the roots. The gases, carbon dioxide entering and oxygen exiting, that is going to happen through these very special openings. They're sort of like pores that are in the leaf surface. And then, of course, we have the formation of the glucose, which is happening deep within any of these cells, and then that's going to move all throughout the plant. So let's take a look at how certain structures of a plant are going to help to dictate their function. So first of all, we're going to talk about stomata. Okay, the stomata, or stoma, those are the openings in the epidermis, meaning the skin or the very surface layer of a plant, that is going to allow gas exchange. So it's going to be a combination or a cooperation between two key cells here. So if you look, what we have here is I have one cell right here, okay, and it's sort of sandwiched up right next to this other one here, okay. These are called guard cells, and the reason they're called guards is because they guard the openings or the pores that allow gas to enter or exit a plant. So what do we mean by that? Let me tell you what's going to happen. These guard cells can change shape depending on how much water they have inside them. So down here on the left, these guard cells have very full vacuoles, so we would say that their cells are turgid, and that's what plants would prefer. When these guard cells become very full of water, they're going to swell and they're going to start bending. They're going to bow out like this. And what it's going to create in the center here is this opening, and that's like a pore, sort of like the pores in your skin, which is going to allow carbon dioxide to get in and is going to allow oxygen to get out. So it's a gas exchange. So the structure of those guard cells and being able to fill up with water and change their shape is going to allow certain gases to enter. When those guard cells are not full of water, okay, when their vacuoles are deflated in a way, we see that there is no longer any opening, and so that's going to sort of close it off, and so now gases cannot enter or exit. So that's really why it's very important to keep plants watered, okay? Part of it is that these stomata need to remain open in order for gas exchange to occur. The next piece of the plant cell, of a plant, is xylem. Okay, xylem is going to be the name of the vessels that are deep within plants that help the plant move water all around. So what did we say again? Remember, we said that water is going to enter through the roots of a plant cell. So it's always going to be moving from root to shoot. It's very important that you understand that that movement's occurring in one direction. So what's going to allow this to happen? Here's a little quick quiz. See if you remember your properties of water. Remember, we're going to have cohesion, and that is going to allow water molecules to be attracted to one another and form a bond. So we say that they stick together. And then we also have adhesion. And adhesion is when a water molecule is able to stick to some other surface. So if we look at this cutout here of a xylem vessel, we see the water molecules moving up against gravity. And do you see how along the side here they're sort of attaching to the surface? That's because they are attracted to this surface. So if they have an attraction to it, would that be a polar surface or a non-polar surface? All right, it would be a polar surface. Remember, polar substances get along well with other polar substances. So the inner lining of the xylem is polar so that adhesion can occur, and then cohesion between the water molecules, allowing them to move upwards against gravity. But we also have another molecule that we need to move around the cell, and that is going to be the phloem is going to be the vessel, which is going to allow us to move our sugar. So once we produce those glucose molecules from photosynthesis, they're going to be produced up here, right? They're going to be in the green leafy structures. But in this carrot, for example, which is mostly underground, these cells of the plant are not green, which means they are not containing chlorophyll, which means that's not where photosynthesis is happening, and therefore they are not creating their own sugar. So what's going to happen is here is where the sugars are going to be made from photosynthesis. Now we need vessels that are going to help transport all of the glucose wherever they need to go. So glucose can move in every which way. The phloem is going to be the vessel that helps them move like that. But remember, the xylem, which moves water, only moves water in one direction. So let's get even more specific. Let's start talking about what is going on inside this chloroplast where photosynthesis is happening. Okay, it's not that we just dump in sunlight, water, and carbon dioxide and poof, out comes glucose and oxygen. There are going to be different phases of this process. And at a high school level, we're going to simplify those phases. If you are interested in learning more details, you would need to take an AP course, but we'll address that later. So let's just get some terminology down for parts of the chloroplast. This whole big silver, bean-like shape here that we see, this is a chloroplast, and then what they've done is they've cut away the side of it so that you can see inside. Because inside the chloroplast is where this magic really happens. The thylakoid, each one of these little ovals, okay, is its own structure within the organelle, and they're called a thylakoid. They're all wrapped within their own membrane, okay? So it's sort of like if you know what Mentos candies are, right? If you would open the wrapper and you see the stack of them all one next to the other, that's kind of what the stack of thylakoids look like. And then they all have a membrane, which would be the equivalent of the candy coating. A stack of these thylakoids is called a granum. And there's a whole bunch of these grana, which is plural. Let me just put that on there for you. Okay, grana is going to be the plural form. There are a whole bunch of these stacks of grana within the chloroplast. Then we have the stroma, which is going to be the space in between. The stroma is the most similar to the cytoplasm. It's not really a structure. It's more the empty space in between other structures. So the thylakoid is going to be wrapped in its own membrane, and this is specifically where light is going to enter. So if I wanted you to answer as narrowly as possible the question, where is light absorbed within a plant? The correct answer here would be that it is the pigment molecules that are embedded within the membrane of the thylakoid, which are found within the chloroplast of certain plant cells. That's about as specific as it gets. Okay, so now let's take these photosynthesis reactions and let's break it down into its pieces so we can understand. One thing you want to keep in mind, and maybe you want to write it at the top of your slides, you want to keep looking back at the overall equation for photosynthesis, because we're going to show you where each reactant is used and where each product is made. So here, this is the whole of photosynthesis. You're going to see we're going to divide it up into two stages. Stage 1 is called the light-dependent reactions, and guess what it requires? Light, yes. So in a nutshell, what's going to happen is sunlight energy is going to be absorbed. We're also going to take in water through the plant's roots. Both of these materials are going to head to the membrane of the thylakoid deep within the chloroplast, and a series of reactions are going to occur. And the products from that, we're going to make 6 oxygen molecules, which we're not really interested in. But more importantly, we're going to make some carriers for energy. And let me explain that in the next slide. All right. So here's a more detailed look at the light reactions. Do not concern yourself with memorizing the details here. I'm simply explaining some things for your understanding. Energy is going to be absorbed and water is going to be taken in. As soon as water comes in, we're going to rip it apart. We're going to break it down into different pieces. From this water, we're going to extract electrons. And electrons are really neat because they're these little balls of energy, and when we take in the sunlight's energy, it sort of like hypes them up, and so they get charged up and full of lots of energy, which is a really good thing. What's left over then? We have some hydrogen ions, and they're going to be used to help move some molecules in these reactions. And then the last thing is the oxygen. The oxygen isn't useful for photosynthesis at all. And so what's going to happen is the plant is going to give that off, and it's going to release that gas. So let's focus on the main event here, these electrons. As the sunlight's energy is being absorbed, we're going to hype these electrons up, and we're going to trap all of their energy in these little electrons. And as we move them through each of these mini reactions, again, you don't need to know specifics here, the end result, this is what you do need to know, is that we're going to generate a little bit of ATP. So we're making a little bit of cell energy, but it's really just enough to get us through these reactions, to help us power through photosynthesis. The main event here, the whole purpose behind what we're doing, is this guy right here. We are making a molecule, and it is called an electron carrier. What do you think it does? Yep, it carries electrons. See. It carries the electrons. So we're going to use the energy from the sun, and we're going to use that to hype up all of the electrons, and then we're going to trap them in these electron carriers. And the reason we're doing this is because we're going to move the electron carriers to stage 2. We're going to move them to stage 2. So the whole purpose of our light reactions, we took in the energy of the sun and then we trapped it in this electron carrier molecule. Okay, we need to find ways to take in the energy and trap it, and then take it in again and trap it, and take it and trap it. If not, then all of the energy would be lost. We'd lose a lot of it as heat. So we're going to trap it little by little and have these small reactions occurring. So just to sum up the light-dependent reactions, it's going to need light. It takes place in the membrane of that thylakoid piece, which is deep within the chloroplast. The reactants are water and light. The products, we make some oxygen, which is given off as a waste product. We make a little bit of ATP, just enough to power the rest of photosynthesis. And the main thing that we are making here, big star, is are the electron carriers. And NADPH is just the name of the electron carrier. It might be interesting to note here, we spent a lot of time in the last unit talking about passive and active transport, and we actually use passive and active transport both during the light-dependent reactions in order to help us make enough of that ATP molecule, just enough to power through the rest of photosynthesis. So now we're going to move on to stage 2. So quick recap, we took in water and sunlight. We generated some energy and some electron carriers from that, and we gave off oxygen as waste. So now what are we going to do with those electron carriers? We're going to feed them into stage 2. And here's basically what's happening in stage 2. We're going to use 6 carbon dioxide molecules, and we're going to break their bonds and rearrange them through a series of reactions. We're going to get the energy to do that from those electron carriers from stage 1. And our end result is that we're going to produce one 6-carbon sugar, C6H12O6, which is glucose as we know it. Okay? So let me just explain this again. Here we have our 3 carbon molecules, okay? And we've ripped the carbons off of the carbon dioxide. We're only interested in using the carbons. We're going to need a little bit of energy in order to power through this reaction, so that ATP that we made in the light-dependent reactions, we're going to spend that here, so we're going to break that down and release the energy. We're also going to release the energy from the electron carriers, so we're going to break down the electron carriers, which releases those electrons, so we have the energy. And basically all that's happening is we're going to make a bunch of different products which convert one into the other until ultimately we get, our end product right here. And what this represents is a 6-carbon sugar, referring to glucose. So we took in carbon dioxide, we spent a little bit of energy, we also released the energy from our electron carriers, and all of that was used to power these reactions, which ultimately made one molecule of glucose. Quick summary, we took place in the stroma. So that means that we were using the thylakoid membrane before, and then the materials are going to move out of the thylakoid and into the stroma, or that extra space in the chloroplast. The light-independent reactions, or we also call them dark reactions, they are carbon dioxide and the electron carriers, which are from the light reactions. If you remember, that was that NADPH molecule. Okay, you don't technically have to know NADPH, but you will hear it referenced a lot of times. So be able to recognize that NADPH is an electron carrier. And finally, we're done with photosynthesis. This was the whole purpose of this big, long shebang. We wanted to make a glucose molecule. So all of that work just to make one molecule of glucose. And remember, the purpose is what we are doing is we are storing energy in this glucose molecule until our cells need it. Okay, so the glucose is sort of like, storing all of your money in a $100 bill to carry it around much more easily, less likely to spend it, versus carrying around $101 bills, okay? So here's just a diagram to sort of, again, go over the overall, all right? So light is taken into the light reactions. We are also going to be taking in water, and from that we're going to produce oxygen, which is given off as waste, as well as making a little bit of ATP energy to help us power through the next phase, and this NADPH, which is our electron carrier, which is also going to go into the next phase. You see the term Calvin cycle here? It's just another name for the dark reactions. Guess who discovered it? That's right, Dr. Calvin. And what is happening in the dark reactions over here, we take in some carbon dioxide. We go through a series of reactions where we break down the energy from ATP. We break down the electron carriers and release their energy. And we use all of that in order to power the formation of our sugars or our glucose at the end. Okay? So that is all the more detail that you need to know about photosynthesis. Make sure that you review your notes. Go back and check the objectives. Most important thing, know the difference between the light and the dark reactions, and make sure that you know what reactants are coming in, what products are being made, and where each of those things are happening within the plant.